Part 2 of Chapter 16 continues on the idea of how some of these microbes are able to infect and ultimately cause damage inside the host. So remember that not all of the bacteria we're referring to can do every single one of these things. Each one's going to have either one or a couple of these different opportunities to cause damage. And so the one we're looking at now is how can we actually deliver the proteins, and when I say we, I mean the bacteria, deliver the proteins designed to actually cause damage into the host cell. And so one of the strategies that's been developed by some bacteria is the type 3 secretion system, also known as an injectosome. And what you see here on the picture is a typical type 3 secretion system. You can see it's gram-negative only because we got two membranes here. And each of these components is an individual protein that all come together to form this syringe-like apparatus that ultimately is used by the bacteria to send the effector proteins directly into the host cell, ultimately to cause damage or death to the cell. Over here we have an actual picture of electron microscope picture of one of these injectosomes. Next, we have to be able to break through the innate immune system, and one of the biggest parts is the skin and the mucous membranes. And so some bacteria are going to rely directly on getting into the onto the other side of the skin by going in through a wound, and so this is how most bacteria want to get in. However, there are other opportunities, such as being injected by a mosquito or tick or flea, such as the Yersinia pestis, which caused the bubonic plague in Europe and we still have issues with here in the United States. Penetrating the mucous membranes is typically going to be part of any sort of respiratory or digestive disease, and so in this case we're ingesting all these bacteria all day, we're breathing them in, and so if they're able to grab hold and adhere to our epithelial cells, well, now they have to actually get brought into the cell to cause damage. And so here's another example of how the type 3 secretion system that's found in Salmonella and E. coli work when it comes to foodborne illnesses. And so here we have a bacteria, and it's injected its effector proteins into these cells, and we can see here this ruffling effect where basically it's causing the cell membrane of the host cell to change configuration, where the bacteria is then able to go straight in and start causing further damage, which will then be signs and symptoms of food poisoning. Now, as we discussed in Chapter 14, we also have the antigen sampling systems, which were salt and malt and whatnot, but here we're showing a picture of the mucosal associated lymphoid tissue. And remember, what we have here is we have the intestinal lining. So here would be the inside of the intestine. Food would be going across here. You have the cilia of the epithelial cells that's basically helping push the food through. And every now and then we're going to have these M cells, which are associated with those malts. And so what's occurring here is that the M cell is just doing a little sampling, testing to see what you've eaten, if there's anything dangerous. And if there is, on the other side, there's macrophages that are designed to destroy it. Well, in certain cases, some of the bacteria that causes disease can actually take advantage of this. They'll go across the M cell, they'll be processed by the macrophage, and then ultimately the macrophage is then destroyed by the cells. So in this case, Shigella is a good example of this. It gets to the other side of the intestinal lining. Once inside the macrophage, it induces apoptosis of the macrophage, and essentially Shigella is able to escape, and then it's able to go cell to cell by going across, causing damage and destruction and death to these cells. Some other ones, like Mycobacterium tuberculosis, are able to get into the macrophage and then ultimately they block the macrophage's ability to destroy them, ultimately causing a stalemate situation. Hiding within a host cell is a very good choice. This is usually something we reserve to viruses, but in some cases some bacteria are able to do it, as we've seen in the last slide. Shigella just goes from one cell to the next once it's actually gotten into the epithelial cells. And another one that causes foodborne illnesses that's been hot topic here in the last year or so is Bluebell's issues with their ice cream with listeria. And so when we get to the digestive system chapter, we'll be discussing listeria and some of its problems that we have as a pathogen. Now, some other opportunities that they have is avoiding killing by the complement system proteins. And so Neisseria gonorrhoeae actually hijacks the host system and binds up all the complement proteins to avoid any sort of a membrane attack complex. So in this case, remember the C5 through C9 come together to form a pore in the bacteria and ultimately cause the insides to spill out through that pore. Well, here Neisseria gonorrhoeae is able to block those complement proteins and basically shut them down to the point where they're no longer able to form those pores. Here, once inside, there's a couple different ways that the pathogens can avoid destruction by the phagocytes. So one is a chemoattractant, C5a. Remember, when C5 is cleaved in A and B, B becomes part of the membrane attack complex, and A is a chemokine. Well, in this case, Streptococcus pyogenes, which we'll see in chapter 21, is responsible for strep throat, but one of the ways that it's able to cause disease is by hiding or camouflaging, and what it's able to do here is it actually degrades or destroys the C5a chemokine, which is going to minimize or at least slow down the attraction of the other immune system cells to the site of infection. Other ones are actually just going to straight up destroy the phagocyte by causing it to die through necrosis or apoptosis. In this case, the same exact bacteria can produce a toxin that essentially kills the phagocyte once it has been ingested. Other ways is capsule. Capsule acts like a as camouflage essentially, so it's not going to allow opsonization. Remember, that's the responsibility of C3b. Without the C3b binding to the surface of the bacteria, there is no opsonization that occurs, and therefore acts, like I said, almost like a camouflage. Streptococcus pneumoniae is one of the key bacteria that we see that is able to do this, and actually, if you remember back to your experiments, Fred Griffith in 1928 used Streptococcus pneumoniae to show that the ones with capsule were virulent, while the other ones were non-virulent that did not have the capsule, and further went on to prove that bacteria were able to transfer the ability to make capsules between themselves, because bacteria are really good at picking up DNA. And so this is a very well-known bacteria that's able to block opsonization. The M protein is one of the things that inactivates C3b, and so some bacteria actually produce what's called an M protein and don't have to rely on their capsule if they're not able to produce a capsule. So again, we're back to Streptococcus pyogenes. The strep throat bacteria is able to bind to different proteins that are going to essentially inactivate C3b. And the last one is one of the ones I enjoy the most when we're thinking about this, is Fc receptors. So essentially on the surface of the bacteria, the variable regions of the antibody should be bound to the surface, and the constant region should be out here as it's shown to essentially allow the rest of the immune system to recognize it. However, certain bacteria such as Staphylococcus aureus and Streptococcus pyogenes actually have Fc receptors on their surface where they basically will bind to the Fc portion of the antibody, thus shutting it down where it's not able to actually be bound up by the immune system, essentially hiding the antibodies that way. Now, they can also survive in the phagocytes by escaping from the phagosome before there's actually a lysosome fusion. So in this case, we have to remember what's actually happening inside that phagocyte. The phagocyte has now brought in the bacteria and puts it into a vacuole called a phagosome. That is essentially just a holding cell, mainly just a, holding cell is probably the right word. It's not until the lysosome, which carries all the toxic chemicals and the enzymes that are designed to just essentially destroy and degrade the bacteria, it's not until the phagosome binds to that lysosome and ultimately forms, as you see in the second bullet, a phagolysosome, that you're actually going to have any sort of mixing of the bacteria and some of the toxic agents that the macrophages carry with them. So there's three different ways that some pathogens are able to survive this. So the first one is just escaping the phagosome altogether before it binds to the lysosome. Listeria and Shigella are pretty good at this, and once they do, they basically destroy the macrophage and get out. Other ones are actually able to block the phagosome-lysosome fusion, and so Salmonella is pretty good at this. They actually produce proteins that block the fusion, as well as Chlamydia also is able to block that fusion of the lysosome. And then there's only really one example of a bacteria that causes disease in humans that is able to survive within that nasty environment of the phagolysosome. And so this is going to be after fusion. In all intents and purposes, this bacteria should be dead, but it does have the ability to protect itself and survive this issue, or survive inside the phagolysosome. Now, they can also avoid the antibodies. So I remember IgA is the one that's important for mucous membranes, and it comes in as a dimer form. So what's occurring here is IgA protease. You should be able to look at that word protease and go, okay, this is an enzyme that breaks down proteins. And so what this one's going to do is it's going to cleave the dimer formation of IgA and ultimately render it ineffective. And so in this case, Neisseria gonorrhoeae is able to do this, as well as antigenetic variation. So remember, what an antibody actually looks for is it looks for the antigen. And so Neisseria gonorrhoeae is very good at actually changing the antigenic structure of its pili, essentially causing the immune system to restart producing the antibodies, because it realizes that what it was producing antibodies for is no longer there. So it's sort of like a chameleon type situation where it's able to change its outside surface to better allow for survival. And Streptococcus pyogenes is pretty good at its capsule that contains hyaluronic acid, which is actually found in our own tissues. And so at this point, the actual outside surface of Streptococcus looks a lot like self, and therefore our immune system sometimes will actually avoid it to keep from actually producing an antibody response against something it assumes as self. Now, when we look at the damage to the host, it can be direct or indirect effects. Direct is typically going to be some sort of toxin-produced type 3 secretion system, something that is intentional, causing damage, where indirect is more just collateral damage based on whatever growth of the bacteria is causing. So in this case, it could be something that's not intentional, but ultimately causes us to have signs and symptoms. Now, damage may help the pathogen to exit and spread. So in this case, we want to think about the bacteria as actually being living organisms, which they are, and all living organisms want to reproduce and continue spreading their seed down through generations. And so in this case, the bacteria don't want to be trapped inside a host that's on the verge of dying. And so something like Vibrio cholerae, which we call the two-bucket disease for those of us that travel, where you essentially need a bucket for your vomit as well as your diarrhea, because you're doing both at the same time, as it shows here, 20 liters a day, which is really roughly about 4 to 5 gallons. And so in this case, with the release of that much fluid, there's going to be quite a few bacteria that are also going to be released too. And so going into sewer system is fine here in the United States, but in other countries where they have inadequate sewer systems, or maybe absolutely no sewer system at all, this ends up being a problem trying to clean up this much waste. And so the bacteria is able to spread very easily in this way. So typically when we have a couple patients that end up contracting Vibrio cholera, it's not out of the realm of possibility that could end up being an outbreak, an isolated outbreak in that village or in that neighborhood. The other one, Bordetella pertussis, is the one that causes whooping cough, and so this is typically found in children that are unvaccinated, and the bacteria gets into the lungs, and as part of its strategy for exit is that it causes the individual to cough with such severe aggression that the child ends up having issues breathing. And so that's where it gets its name. Whooping cough is the sound that the child makes when it's finally able to catch its breath and be able to catch a breath, but then continuing to cough with such aggression that the respiratory droplets are shooting pretty far away from the child in hopes that it's actually going to be picked up by another individual. So toxins come in two different types. We got exo and endo. Exotoxins are ones that are actually secreted by the bacteria. There's also a little caveat here that they can actually leak out of the bacteria after the bacteria is killed. So if the bacteria lyses in any sort of way, some of these toxins can actually leak into the tissue and still be just as effective as if they were secreted. So foodborne intoxication is one of the bigger ones. Tend to find Salmonella, E. coli, Shigella all produce toxins, as well as a litany of other digestive diseases that we'll see in chapter 24. And one of the best things about exotoxins is that they mostly are heat sensitive, so we can destroy them pretty easily with heat. And so this tells us that most of them are protein based, because proteins end up getting three-dimensional shapes, and those three-dimensional shapes can unravel pretty quickly with heat. So that's why these toxins are heat sensitive. Now, some toxins act locally, which means that, again, like something like the flesh-eating bacteria is going to be locally producing toxins that's helping it chew through the epithelial and the muscular and connective tissue, where other ones are going to be released and actually get into the blood system and go septic. And this is going to be the worst case scenario for many of these toxins, is if they get into the bloodstream, then it becomes really difficult to actually get the patient under control and before the toxin causes death. And that's going to be the biggest issue here, is that many of these are fatal before the immune response can get mounted. You remember, in order for a full immune response, it takes anywhere up to 2 weeks. And if you have toxins being produced that are actively damaging at that moment, you don't have time to wait for 2 weeks for a strong antibody response. So this is why vaccines are critical. A lot of these toxin secreting bacteria, there are vaccines available for. And so this is the route that we always want to take. And most of these vaccines are actually toxoids. Toxoids are just inactivated toxin. The toxin's been heated up, it inactivates. But again, the amino acid sequence is all still there, so the immune system is able to mount a very strong antibody response against that toxoid. So in case we ever see that particular bacteria again, the toxins will be quickly recognized and destroyed. Now, if a patient comes in that hasn't been vaccinated, or maybe their vaccine titer levels, antibody titer levels aren't high enough, we do have availability of antitoxins, which are essentially just antibodies that have been isolated to treat people that have been exposed to these toxins. And so, very much like a snake bite, we have antivenom. Here's antitoxin for the toxins produced by the bacteria. These toxins, these exotoxins, come in 3 different styles. Neurotoxins damage the nervous system, enterotoxins are going to damage intestinal, and cytotoxins are just basically catch-all for any cell type. So here's some tables. I won't go through them, but tables of different diseases and the toxins that actually produce. And that goes on for 2 slides here. Now, the exotoxins mainly come in a style that's similar in all toxins, but they get called AB toxins, where the B portion is the one that binds to the cell receptor. These are very specific, so if the B portion only binds to epithelial cells, well, then it's not going to cause any damage to nervous system or any other type of cell types. It's going to be dependent on what the B subunit recognizes. And so this allows for some really good strategies for pharmaceuticals, in terms of the B subunit is so good at being able to bind to receptor that all we have to do for some cases in pharmaceuticals is remove the toxin and actually put a pharmaceutical medicine on there. And we've been having some pretty decent success with some of the newest generation drugs coming out that take advantage of this. Now, once it's inside the phagosome and it has a lysosome fusion, those actual enzymes cause a break between the B and the A subunit. And the A subunit is then actually the toxin. It's actually an enzyme that traditionally has some sort of negative reaction to our body in terms of causing damage. And so the A subunit's always the toxin, where the B is the cell receptor. Another type of toxin are membrane damaging, and so these are going to be ones that damage the plasma membrane. In case of some of them, hemolysins are able to lyse the red blood cells. So we'll see in chapter 21 that Rebecca Lansfield created the Lansfield groupings, and one of the things that we see there is that the red bloods, that those plates are what we call blood auger plates. And so the blood is coming from sheep. It's 5% sheep's blood, and certain variations of Streptococcus actually produce a toxin that destroys the red blood cells and actually produces a clearing around the colony when grown on these Petri plates. And so you'll see that in chapter 21. And so that's a toxin called hemolysin, essentially just a, as you can see from the name, hemo's blood, lysin means to cut, so we're cutting up red blood cells. Some insert into the membrane and actually form pores. So streptolysin from Streptococcus pyogenes actually causes the host cells to burst by just poking holes in them. And other ones are able to destroy the phospholipids by having enzymes available called phospholipases, which are going to destroy the phospholipid bilayer of our host cells. And this is something that belongs to a bacterium that we'll be talking about in the digestive as well as the wound called Clostridium perfringens. Another variation of exotoxins involves what they call the superantigens. And so in this case, we have to remember back to chapter 15 when we're talking about how a T cell gets activated. And so in this case, it has to have the antigen presenting cell, whether it be dendritic or macrophage, bringing the antigen inside the MHC class II. And remember, the T cell, the CD4 T cell, has two jobs. One, it has to recognize the MHC class II, and then it also has to recognize the actual antigen. And if both of those are not done together, then there is no activation of the adaptive immune response. Well, in this case, some toxins actually cause the T cell to become activated even if it doesn't recognize one or both of the things inside the MHC class II. And so what occurs here then is the T cell is put into hyperdrive and starts producing all kinds of cytokines unnecessarily. And this a huge release of cytokines actually causes what's called toxic shock syndrome, which essentially these cytokines, what's their job? Their job is to dilate the blood vessels. Their job is to initiate inflammation. Well, if these actually get inside the blood vessels and spread throughout the body, causing dilation of all the blood vessels, essentially you're going to bleed out internally and die of shock due to low blood pressure. There's several other different types of toxins out there that get put into all kinds of different catch-alls, such as exfoliatin causes what's called scalded skin syndrome in children. It's usually where the bacteria is producing this toxin that causes the skin to separate from the basement membrane. And this allows the bacteria to travel throughout the body, but ultimately is causing the skin to look like it's been burned off of a patient. Other different types of toxins can come in all these other flavors that are highlighted at the bottom. Proteases, enzymes that break up proteins, lipases, enzymes that break up lipids, collagenases, so forth and so forth, to help the bacteria ultimately survive within the host. Now, there's only one endotoxin, but there's a large cohort of bacteria that are able to produce that endotoxin because that endotoxin is LPS, lipopolysaccharide. And the only time we see LPS is in gram-negatives. So essentially every gram-negative bacteria produces endotoxin in this fashion. It's usually at the tip, so go back to the chapter where we discussed, I believe it was chapter 3, if I remember correctly, discuss the makeup of the gram-negative bacteria and specifically the LPS. And so you'll see that the LPS is made up of 3 things, but here we're mainly focusing on the lipid A, which is found on the surface of the LPS or at the tip. And by having it there, it's going to be the first thing that the immune system recognizes, and it's going to actually trigger a huge inflammatory response, followed by cell lysis, and then ultimately activation of the innate and adaptive, further causing the inflammatory response to be heightened. And therefore we end up having the same problems with systematic shock as we did with the gram-positives. One of the biggest differences between endotoxin and exotoxin is exotoxins are secreted, while endotoxins stay with the bacteria. Again, the bacteria didn't produce the LPS to be toxic, it just so happens to be something our immune system does for us. And so the other big difference is that endotoxins are not heat. They're not going to be destroyed by heat. And so autoclaving does not necessarily destroy gram-negative, the LPS. And so we've actually done studies in mice just injecting the LPS only and causing the death of the mouse. So even if the bacteria are dead, if the LPS is still there and still recognized by the body, it could potentially cause death even without a bacterial infection. So, as we can see here, exotoxins can be from both gram-positive and gram-negatives. They're heat inactivated pretty easy because they're proteins, so they make three-dimensional shapes. The endotoxins only come from gram-negatives, and this is a saccharide. It's a sugar, LPS, lipopolysaccharide. So it does not have a three-dimensional shape, so therefore the heat is not very useful against it at all for degradation like it is in the protein exotoxins. So damage into the host is also going to be the responsibility of our own immune system. Our own immune system sometimes gets out of whack and starts causing too many problems, such as toxic shock and whatnot. And so sometimes if it's a virus, such as the virus that causes chickenpox and shingles, there's really not a whole lot we can do if somebody comes in with an outbreak of shingles right now with current technology. So instead what we can do is give them a prescription of prednisone, which is a steroid that's going to slow down the immune system's response, kind of make it a little less excited about what it's trying to do. And by doing that, we can actually get healing to happen a little bit faster, also a little bit more comfortable for the patient if their immune system is truly just acting out. And ultimately what we're going to be able to minimize is any sort of damage that's associated with inflammation or the adaptive immunity. So examples here of adaptive immunity is that once the antibodies are produced and they start binding to their antigen, they can start falling out of solution. It makes them less soluble. And so by doing that, they fall out of solution. They end up landing in places like your joints. And if this occurs, it actually gives very similar arthritis, to arthritis conditions in that patient where they're having serious trouble with their joints. And another place that it can get caught up is in the small ducts of the kidney. And so if you have antigen antibody blocking the ducts, it's going to slowly cause the damage to the kidney and ultimately may cause the kidney to die. So we need to be careful with that. Other things are that the antibodies, since they only recognize epitopes of 10 to 15 amino acids, there could be a very, very remote small chance that those same amino acids are found somewhere in our self. And so very rarely, but it does happen, we have cross-reactivity with antibodies produced against an infection that actually also recognize self. Now here we're switching strategies a little bit, and we're going to viral pathogenesis. And so when it comes to viruses, their main goal is to hide inside our cells, take over the machinery of our cells, reproduce themselves in incredibly high numbers, and then get out. And so here the very first thing that needs to happen is the same thing that needs to happen in bacteria. We're going to have some sort of binding to the host cell. We're also going to have that virus now, instead of adhering as we did with bacteria, we're going to have to invade. That virus has to get inside the cell in order to survive and actually reproduce. So the virus is going to attach to target cells. So in this case, HIV targets CD4. So the receptor CD4 that happens to be on T cells is the same receptor that the HIV virus is looking for. And so once it sees it, it's going to bind and it's going to be taken up into the cell. If the HIV virus binds to any other cell type, it is not going to be taken up if it does not have that CD4 receptor. So once inside, it's actually going to be hidden from any of the external immune system capabilities. But remember in chapter 14, one of the things we talked about was interferons, where essentially they have the ability to block viruses on the inside of our cells. And so here, while that's a great strategy for us to produce these interferons and ultimately try to slow down virus replication, the virus also has another counterattack. It's able to block sometimes the expression of these interferons. Now, viruses are able to avoid the antibodies by just staying inside the cell. The antibodies are not found inside the cells, and so any antibodies that might be floating around, the virus is going to be fine as long as it's hiding inside the cell. Now, occasionally the virus needs to break out of the cell and then go and infect other cells, or even better, some of them are able to cause cell fusion of two host cells together, and now it doesn't actually have to leave the cell. It leaves the one that it's already replicated in and gets into the next one and starts replicating in there, and thus never actually had to see the outside of the cell and potentially be taken up by the antibodies. So here's some examples of some other ways that the virus can trick the immune system. So one of the ways is to prevent or delay apoptosis. The big protein that's responsible for deciding if a cell is going to go through cell suicide or not is p53. And so in this case, the virus can actually block p53's ability to decide for apoptosis, thus surviving inside the cell for a few more minutes or hour, whatever it may be. Other ways is to block the class, MHC class 1. So for those of you that didn't do too well on the last exam, remember that the MHC class 1s are found on every single nucleated cell. And so by having that, it becomes a very good flag for, hey, I'm self. This is a normal cell that has an MHC class 1, so please don't attack me. Well, if the cell ends up being infected, it will take part of the virus and put it up on MHC class 1, acting in distress for the CD8 T cell to come over and destroy it. Well, that's no good for the virus, so the virus is going to put up a fake MHC class 1 and try to play it off for as long as it can. But ultimately what's going to occur is that the NK cells, the natural killer cells, are going to show up and destroy that cell. So depending on how much time the virus has bought by playing this little game of camouflage and hide and seek, it may actually get to the level where it's replicated high enough that it doesn't matter. That cell's going to be done anyways. So the last part is eukaryotes, and so as I've said before, I don't really pay much attention to the eukaryotes because most of these do not cause serious disease. Most of these are perfectly fine microbes that are able to survive on their own out in the nature. They don't need us. They don't need to survive in us whatsoever. And so the biggest thing is that most of these are actually opportunistic pathogens, meaning that in most of us, we're never going to have to deal with these. And so this is by no means a all-inclusive list, but it gives you an idea of some of the more popular ones, such as Candida albicans, which is the yeast that's involved in yeast infections, Aspergillus fumigatus, which is basically black mold, and Cryptococcus neoformans, which is a yeast that's found in pigeon dung, but we're always breathing in when we're downtown or wherever there may be large pigeon cohort, so most of us are breathing in Cryptococcus neoformans without even knowing it. So the biggest problem with funguses and eukaryotic pathogens is going to be in the immunocompromised people. Cryptococcus neoformans is one of the top 10 leading causes of death in HIV patients. Now the protozoa and helminths, also eukaryotes. Helminths, remember, are worms, where the protozoa are going to be more microbes, but both are eukaryotes, and so they have some ways to hide from our immune system, but mainly to sit there and not really cause much problem in terms of being serious life-threatening diseases. Alright, well, hopefully this helps, and if you have any questions, please don't hesitate to email me. Thanks a lot for listening, and we'll see you soon.